Biology Genetics Notes, Part 1. A couple of genetic facts for you. In certain isolated communities, 1 out of 100 boys is born with a genetic defect that causes muscular dystrophy. Intermarriage causes the disease hemophilia is spread through the royal families of Europe, almost wiping out certain families. The first genetic research lab was actually a monk's abbey garden. And the same genetic defect that causes sickle cell disease can also protect you against malaria. Gregor Mendel is known as the father of genetics. He was an Austrian monk, and part of his job in the monastery was to work in the garden. In doing so, he started working with seed plants and developing our modern genetics theory. Genetics is the study of heredity. Heredity is the study of passing of traits from parents to offspring. Mendel chose pea plants as his experimental subjects mainly because they're easy to cross and because they showed a variety of contrasting traits, such as purple versus white flowers, tall versus short stems, round versus sprinkled seeds. There are 7 different traits that he focused on with pea plants. This diagram shows them. So again, we have flower color, flower position, seed color, seed shape, pod color, and stem length. Cross-pollinating plants, and a little mini lesson for you. Plants have male and female parts. In this diagram, we can see on this left side, this big part here is the female part of our plant. Okay? Right here is our female part of our plant. These little stamens coming off are the male parts. They release the pollen. The pollen is what fertilizes our flowers, creating more flowers. Plants can either do this on their own, or researchers can cross-pollinate and force it to do what we want. So what Mendel did on the right is he took this purple flower and he cut off the stamens, cut off the male part, and then he got the pollen off of this white flower and dusted it over the ovary of this purple one, thus pollinating or fertilizing this flower with the white pollen. Okay? As it matured his he's able to plant seeds, and he created his first generation of offspring. That is what cross-pollinated is. In Mendel's experiment, for example, he crossed our P1 generation, which is the parents, red flowers, by white flowers. The first generation of offspring we call the first filial generation, or F1 generation. And in this case, they had all red flowered offspring. He then was curious about what happened to the white flowers, and he took those white offspring from the parents and he crossed them with each other. The second generation, the F2 generation, or the second filial generation, produced 3/4 red flowers and 1/4 white flowers. The white flower reappeared after skipping a generation. From this, Mendel came up with the modern genetics theory that we still use, and this occurred hundreds of years before we knew what DNA was or how DNA works. To show you what Mendel's looked like with our Punnett squares. So our parents are on the right, okay, and so here's the red parents across the top with the capital R's. Our white parents are on the side with our 2 little r's. And all of their offspring are big R, little r. He then took 2 of their offspring and crossed them on the chart on the right. And so we see we have the big R, little r, big R, little r. And when he crossed those, we have his 3 red plants to his 1 white. And here's a different format. Again, we crossed, in this case, we have purple by white. The first filial generation, we had all purple plants. We crossed those by themselves. We had the 3-to-1 ratio, purple to white, in our second generation. So what Mendel figured out, or his analysis, stated that F1 generation always displays only one trait. He started calling this one the dominant trait. The first generation must also have within it the trait from the original parents. That white trait must be there, it's just not seen. He then called that one the recessive trait. It's hidden. So when he crossed the F1 generation by itself, our F2 generation displays that hidden trait in the ratio of 3/4 of the dominant one to 1/4 of the recessive. He also determined that each individual has two factors, is what he called them, that determine what paternal appearance the offspring will have. Mendel developed three laws or three principles of inheritance. The first is the principle of dominance and recessiveness, and that one gene can dominate or mask the other. The dominant gene is always the stronger gene. It's always expressed if it's present. It's going to take over. We use a capital letter to represent our dominant gene. For example, with red flowers, we use a capital R, or for tall plants, we use a capital T. With recessive genes, it's weaker. If a dominant gene is present, it's going to hide this. It's expressed only when we have no dominant gene present. Most of the time, we have two of these. So we use a lowercase letter of the same letter we use for the dominant gene. So in this case, we have a little r, which is recessive, which shows the white flowers, or a little t to show that short plants is recessive to tall. Mendel's next principle is the principle of segregation. This occurs during meiosis. We know that each egg and sperm cell has only one gene for each trait. Remember, they're haploid, they have one of each. And so the genes for traits are going to segregate or separate as we go through meiosis when we form our gametes. So here we have our cells going through meiosis. You can see the parent genes from the parent cell, which there's two here. As we go through meiosis, they split apart, they segregate, so that each egg or sperm only has one of each trait. Okay, just showing you in different ways. Remember the cells here is diploid. It goes through a process of meiosis. We have our haploid gamete. Now, if our gametes go through fertilization, egg meets sperm, for example, we create a diploid zygote, and now it has one of each. If we didn't go through meiosis, we would have too many chromosomes. In a future lecture, we'll talk about why that's not a good thing. Our last principle is the principle of independent assortment. Factors of a trait will separate independently of one another during gamete formation. So as our homologous chromosomes pair up during metaphase, it's independent of the other pairs. For two genes to sort independently, they must be on different chromosomes. So here we have our visual. So here we have these two cells, both in metaphase 1. Notice the cell on the left, we have the red one on top here. But on this one, the red one sorted itself to be on the bottom on this chromosome. The next time they line up, notice in this case, both reds stayed on top. So having to line up is independent of one another. Just because this one forms this way doesn't mean this one has to. It can switch places. Same idea. If I ask my class to get up and form a single file line, it would be in one formation, and then tomorrow I can say, let's do it again. We may not have the same order. It's the principle of independent assortment. So what do we know now? Looking a couple hundred years ahead of when Mendel did his work. We now call Mendel's particles or his factors, we call them genes. And we know that our genes are contained on chromosomes, which are made up of DNA, deoxyribonucleic acid. We know there's alternative forms of genes. We call them an allele. For example, there's a gene for a hair color. We have a brown allele, a blonde allele, a red allele. The alleles are the different forms. We know for each characteristic an organism has two copies of the gene, one from mom, one from dad. We also know that gametes carry only one allele for each inherited characteristic. That's our principle of segregation. And we know that alleles can be dominant or recessive. So looking at our big picture again, we know chromosomes are made of DNA. We know that on these chromosomes we have genes. Those genes will code for different proteins that are formed. We know that alleles are possible forms of a gene, and they come in pairs. The genotype is the combination of alleles for a trait. Okay, it's the pair of genes. If I ask for the genotype of an organism, you're going to give me the letters that we use. And then we can have homozygous or heterozygous genotype. Homozygous, where homo means same, so this is the genotype of two identical alleles. So a homozygous genotype could be big R, or little r. You can also call these purebreds, so they're the same two traits. Our heterozygous genotype has two different alleles. Hetero means different. So in this case, we have big R, or big T. We call these hybrids. They have one of each trait. If I ask for the phenotype of an organism, I want to know what does it look like. Okay, it's the physical appearance of the organism. The phenotype is the expression of the genotype. So, for example, brown eye is our phenotype, or red flower, or that we have blood type A. This is just an example of showing you a chromosome. The top chromosome is our dominant chromosome. It has the dominant alleles on it. I'm sorry. It has the dominant alleles for P and for B. And then bottom one, we have recessive alleles for A and B. The capital letter showing our dominant trait, and our lowercase letter showing our recessive trait. So for our gene here, which is P, it's homozygous dominant. We have two capital P's, so this cell will be homozygous dominant. Here we have homozygous recessive, and two little a. And then for our last trait we're looking at on this chromosome, we have capital B, lowercase b. It's heterozygous for the trait. So once we look at this as a quick little snapshot and summarize what we just learned, we can look at the flower color. So we have two alleles. We could have the allele of red or the allele white. Okay? It will always tell you which one's dominant. So in this case, we have red as our dominant gene. White would be a recessive allele. We can represent the dominant one using capital letters. And the dominant ones, we can have two genotypes. We can have a heterozygous genotype, big R, little r, or it could be homozygous, big R. For our recessive trait, we only have one genotype, which is homozygous, little r. And then remember, the phenotype is just what it looks like. So for our dominant trait, it's going to look like a red flower. For our recessive trait, we'll have white flowers. We'll do one more. For seed color, we have two colors of seeds. We have green seeds and yellow seeds. In this case, green will be recessive, yellow will be dominant. For recessive, we know it's a homozygous genotype, so little y. We're using Y because yellow was our dominant trait. And for our genotype for yellow, we have two. It can be heterozygous, big Y, little y, or homozygous, little y. Our genotypes again, we can have green, or we can have yellow. On the bottom is just another chart to show you the same idea for seed type. Same time. Our last thing in our notes today is we're going to learn how to do a monohybrid cross. If you're still struggling with these, please look at the other videos showing you more examples from our worksheet. For monohybrid cross, we're crossing one pair of contraction traits. We will use a Punnett square to determine the probability of a certain type of offspring having the alleles of the parent. So for example, here we have a tall pea plant with a homozygous versus a short pea plant, which is also homozygous but recessive. This is our pea generation or parent. And so you put it across the top one plant. Here's a tall trait for that plant. Here's this other tall trait. On this side, we put one little t, little t. We fill in our chart, and our offspring will be all big T, little t, which is our hybrid. And they're all tall for their phenotype. If this was too quick, we'll break it down right here for you. So here we have our parent generation. We're looking at color. So yellow will be dominant, and little y will be recessive and green. So here we have two parents. We have a heterozygous yellow parent, so it's dominant with the big Y, little y. We're going to show that demonstrates why it's yellow. And we're going to classify a homozygous recessive green one. Okay, so show the recessive trait. Remember our principle of segregation. So when this parent forms its gametes, it's going to separate its two alleles. It can give a gamete to a big Y, or it can give a gamete a little y. Same thing for this parent. So there's a segregation when it goes through meiosis and forms its gametes. We'll have either a little y, or it can give also a little y. We fill in our Punnett square. One parent will go along the top. So we're going to put this parent across the top, so there's our big Y. And then we'll put his other Y in the next box next to it. Remember, we're segregating, so each letter gets its own box. So then we'll put our next parent along the side. So we have a little y and our little y. Please make sure if you write these that we can tell if your y's are capital or lowercase. So now we fill in our square. Our big Y will come down and fill this box. And this little y goes across and comes in the box. The big Y will come all the way down to the bottom left one. And our little y will come across and go here. We're showing all our combinations. This little y can go in, and this little y can go in there as well. Same from the bottom. The y comes down, and the y goes across. So here are our possible combinations. A lot of times you'll be asked, what is your genotypic ratio? Here you look at the genotypes we have. We have two. We have big Y, little y, and we have little y, little y. So we have 1, 2, big Y, little y, 1, 2, little y. So it's a 2 to 2 ratio. For phenotype, I want to know what color. What colors do we have? So we have, these that have the dominant traits, they are yellow. So we have two yellows. And we have two that are homozygous recessive, meaning they're going to show up as green. That is it for this section of the notes. If you need help, please again look at the other videos showing our Punnett squares, or please come in for help.